Welcome. I'm glad you're here. I'm Jean Lightner, and I'm going to give you a summary of the Creation Research Society's eKINDS research, particularly highlighting its predictive success and evidence leading to new prediction. eKINDS stands for Examination of Kinds in Natural Diversification and Speciation. Wow, that's a mouthful. Examination makes sense. We're biologists, and we're interested in plants and animals and understanding more about them. But when it comes to the word kind, someone in the audience may be thinking, what do you mean by kinds? Well, I'm glad you asked. In the book of Genesis, in the first chapter, we are told that God created different kinds of plants, trees, and other vegetation with seeds according to their various kinds. And that was on day 3 of creation. We are told that on day 5, God created creatures that live in the water and creatures that fly. And this again was according to their kinds, or sometimes it's worded according to its kind. Each of these creatures was to increase and fill the sphere that they were created to inhabit. So, for example, creatures that lived in the water were to reproduce and fill the waters, and creatures that fly were to increase on the earth. On day 6, God created livestock, beasts of the earth, and creatures that move along the ground. And you may have guessed it, according to their kinds, or sometimes it's worded according to its kind. So when we're talking about kinds, we're talking about the creatures that were created at the beginning. And we can see from this that not all creatures share common ancestry. God created different kinds of plants and different kinds of animals at the beginning, and we're interested in that. I'd like to look at one species of plant that we see in our world today, which is descended from one of the created kinds of plants that God made at the beginning. It's Brassica oleracea, and there you see a picture of wild cabbage. Well, this is the same species, and cabbage and collard greens and kale and broccoli and cauliflower and Brussels sprouts are all a single species of plant. Do you see that diversity? That's astounding. We also have variation in dogs. Of course, all dogs are the same species, but they differ in such things as coat color, coat texture, coat length, and other features, different proportions. All this variety has shown up within a few hundred years. We have hundreds of dog breeds, and they have arisen in hundreds of years. Look at that diversity that's showing up. That's amazing. And then there's cattle. Well, we have thousands of cattle breeds that have been developed in thousands of years, and some are very well locally adapted. And you see that type of variation again. That's astounding. And the question that burned in me was, how do they do this? Well, the secular view has an explanation, and it's that random mutation and natural selection basically brought about everything we see. And that sounds rather magical and fairy tale-ish, but the biblical view is a little different. It's recognized that there was some created diversity. Different kinds of plants were created at the beginning, and different kinds of animals were created at the beginning. And God could create diversity even within a kind. But then we have typically used random mutation and natural selection as the reason explaining this diversity that arises within a kind. And I once did. But the more I thought about it, if we randomly change something that's well designed, what do we get? Usually we get a disaster. And I was trained in agriculture, so we did a lot with artificial selection. Artificial selection is very powerful. Dairy farmer can use dairy herd improvement data and select sires, and he can take his cows that maybe are producing milk at a mediocre level, and he can dramatically increase it through bringing genetics in from outside the herd of his choice to help his cows do more with what he wants them to do. But the more I looked at things that were attributed to natural selection, the more I realized, wait a minute, there are limitations to what a farmer can do with artificial selection. I had courses that discussed what's realistic to expect and what you should not expect from artificial selection. And natural selection was being credited with far more than any farmer could ever do, even with his intelligence. So I was suspicious something else was going on. The eKINDS project was initiated in 2016 by Dr. Kevin Anderson, who at that time was the research director at the Creation Research Society's Research Center, and myself, and I was serving on the CRS board at the time. And the goal was to answer several questions. One of the questions was, how did we get the wide variety of today's species from the small number of animals on the ark? Clearly, diversity shows up over time, and we've seen that. We've documented it. How did this happen? How do new species form, and how does this fit within the biblical creation model? Can we trace the spread of created kinds from the ark to where they live today? And for this research, we had other researchers joining us. John Ahlquist was a famous ornithologist and molecular biologist. If you're familiar with bird taxonomy, this is the Ahlquist of Sibley and Ahlquist taxonomy fame. Decades ago, they worked with DNA hybridization, and that was an early molecular technique. Essentially, what happens is you take DNA and you unzip it. And then you put this unzipped piece of DNA in with the unzipped pieces of DNA from a different organism, a different species. And you see how well it zips back up. The more similar they are, the better it'll zip back up. And the more different they are, the less they will. So that was an early technique, and we learned a lot from that. And at the time, he was a theistic evolutionist. But later in life, John became a creationist, and he was very excited about ornithology, and he wanted birds to contribute to creation science because he knew birds had contributed so much to our understanding of the natural world in other areas. I had some great talks with him. He was a lot of fun to talk to. We'd do email exchanges, and he talked about Ernst Mayr, a famous evolutionist that had come up with this idea of a founder effect. And he talked about the different classes he had and the various variations on that theme that he had learned about. And through that discussion, we ended up with this paper, Founder Events, Foundational in the Rapid Post-Flood Diversification. There were a number of things we learned when we dug into the topic, and some of them are here. The founder effect is evolution is considered to be always random. That's the way Ernst Mayr proposed it, but it's not always random. Animals can often choose their environment. And as an example, when the Grants were at work on the Galapagos Islands, the large ground finches would sometimes visit the island. They didn't have a colony there at first. Eventually, over the years, some stayed, and there was a population established on that island where they were studying. But they made it very clear that those that chose to stay were not a random sample of those that visited. And anyone who watches birds and other animals should recognize that, yes, animals can choose their environment. But evolutionists will miss this and assume that the non-random pattern is from natural selection. In fact, a non-random pattern is automatically called selection. That's just the way it does. No one is required to demonstrate that natural selection was going on. It's just assumed because that's the explanation that's always been used. So we need to be aware of the differences, and there was a lot of theoretical work, and it was very interesting to me that theoretically, when a few animals carry beneficial alleles into a new environment, subsequent inbreeding should increase the chance of speciation. And this makes a lot of sense. So if you imagine back at the time when the flood had ended and all those animals were getting off the ark to go fill the world again, those that could were going to go find a place where they liked it and choose an environment they were comfortable in. And they would naturally carry in those beneficial alleles because they were choosing the environment they liked and they were well suited to it. So by God giving his creatures the ability to choose a suitable environment, you had it set up for speciation to occur. And there's probably a lot of it that occurred on a big scale in that time frame. John and I put out several other papers. We did one on paradise kingfishers, and those were the birds that Mayer had been looking at when he proposed the founder effect. We talked about them, the founder effect, and creation research in that paper. There was a series of three papers we put out called Strategies for More Clearly Delineating and Characterizing and Inferring the Natural History of Barrowmans. And that's a mouthful. What do we mean by strategy for more clearly delineating barrowmans? Barrowmans are another word for created kinds, and we wanted to talk about establishing barrowmanic status with application to the order Galliformes. So we just talked about what do we need to do to establish which organisms belong to the same created kind. And let's look at this order, Galliformes, which are your chicken-like birds, your turkey and your grouse and your chickens and pheasants. So we looked at them and we did it with them. Characterizing, what's up with that? Well, we looked at what we decided was in that created kind, and we looked at the diversity, and we tried to evaluate it and see what types of traits are changing and how are they changing. And then inferring the natural history of barrowmans, well, this is like tracing them maybe from where they came off the ark. So our third paper was evaluating relationships and proposing post-flood dispersal with application to the order Galliformes. And in these papers, we put out a lot of theoretical content of this is how we should approach this, and then we basically did it with this particular order and looked at it in depth. So these are foundational that other researchers can use as they apply it to different groups of plants or animals. A second researcher who was involved in the eKINDS project is Matthew Cerati. He's Hungarian-American and fluently speaks more than one language. He has bioinformatics experience, which was tremendously valuable. And he has many creation publications. He's been publishing in the creation literature for a very long time. With him, we wanted to identify created kinds. And I want to back up and I want to look at this topic a little bit to give you a little bit of background. How can we know that creatures were created according to their kinds, but how can we identify which ones belong to the same created kind today? Many created kinds have been tentatively identified, and there's a lot out there in the literature. I'm only going to point to a few papers. In the quarterly, we had a paper on the current status of barrowmanology, which summed up the research that had been done to that point. And there's been lots of research after that. There were a series of Answers Research Journal papers. The ones on birds and mammals were contributed by me, and the ones on reptiles and amphibians were contributed by my colleague, Tom Hennigan. Basically, before Answers in Genesis built the ark, they wanted some kind of estimation of how many kinds there were. And we make the point that these are estimates. We have not gone in enough detail to be certain about them. There are several tools we had for identifying which animals belong to a created kind. Hybrid data is the most important because it takes a tremendous amount of continuity between creatures for reproduction to work. It's really easy for things to show up so it doesn't work. So if things deteriorate, you don't get the hybridization, you don't get the reproduction. So hybrid data is kind of the gold standard, but we don't always have it. Where we do have it, we feel very confident that the two creatures belong to the same created kind, and that's been discussed elsewhere in the literature of why that's such a strong inference. We also used the cognitum, which means we kind of looked at how these animals looked, and we looked at how they were classified. So if there are two genera in a family where we have hybrids, then we're going to assume that all the other ones that fit in those genera are also part of that created kind. So that can be helpful to use their taxonomic placement and appearance. Finally, there's statistical methods that we use. And I want to say something about this. Some people think that if you do something with math, that suddenly makes it true. But whenever you apply math to science, you must make assumptions. And when the evolutionists make their phylogenetic trees, they are using an algorithm that will give them a tree regardless. It's going to give them a tree because that's what it does. In baraminology, we use clustering techniques to find groups. And sometimes things group well and sometimes they don't group well. But we're looking for groups, and whether or not we've inputted data that will give us a result that tells us whether or not they're in the same created kind is always open to some question. So using statistics doesn't make it God's truth. Using statistics is important. We need to be doing that. But you need to recognize these are not perfect methods that give you the right answer every time. We don't live in a world where we have those options. So Matthew, under his pseudonym Genomics, published this paper, Baraminology Classification Based on Gene Content Measurement. And we decided as part of the eKINDS project that we wanted to develop this gene content method. It's similar to some things that had been done in the secular world, and Matthew recognized, hey, this would be good for us. We've used it, and in the process of using it, we have found out some things that will help us use it better. One, we need complete proteomes. We need a catalog of all the proteins that the organism produces. If you go on partial data, you're not going to get good clustering. You're not going to get results that you can really interpret well. Second, for any particular group you would expect, there need to be 3 or more organisms. It's hard to group things when you just have one. It's not really a group. And 3 or more organisms allows us to do various statistics as well. Well, we applied this to data that was out there in the insect orders of Hymenoptera and Diptera. And when we looked at Hymenoptera, they all grouped together in one very strong group. When we looked at Diptera, it looked more like 2 groups. And this was very controversial. The reviewers did not like it. So it remained unpublished, but I did mention it in my previous ICC paper. There was concern expressed that one can keep grouping until everything is included. You can just keep going up and up. But massive gene gains must be invoked to explain the origin of an order in insects. And here we have a picture out of a secular publication that probably used a lot of the same data we did when we ran our evaluation of it. Along the bottom is time. Going up the right-hand side is log complexity. And you will see that all of a sudden complexity goes up very rapidly, and then there is a gradual decline. And then it goes up very rapidly again, and there's a gradual decline. And so basically what they're saying is you have this miraculous, seemingly miraculous time in the past where all this complexity, new genes that are connected together, that work well together, all of a sudden they all show up. And then the rest of the diversification within that group can be explained by a slow loss over a period of time. Well, this means there's a problem when you try to group insect orders together because different insect orders use a very different suite of genes to do what they do. So it's much easier to fit this in the creation model where, okay, orders were separately created. They had to have been because the data really doesn't make sense otherwise. We did use the gene content method on humans and primates, and we had some other animals in there. And we published this paper, The Uniqueness of Humans is Clearly Demonstrated by the Gene Content Statistical Baraminology Method. And if you would like a paper that shows the profound difference between humans and apes, this is one of several that I would recommend. It really showed powerfully that humans, which were represented by humans, Denisovans, and Neanderthals, are profoundly different from the great apes and all primates. All primates actually grouped in one big group, and the great apes were not beside the humans. Well, let's get back to some of the questions that I was interested in myself. Where does all this diversity come from, and why does it seem to just show up? I've been looking at genetics for a number of years to try to figure out the answer to that. I've looked at coat color in mammals, and there's 3 different genes I looked at. Very interesting, very informative. I looked at variable genes in dogs, and there's a list of them there. Some of them have to do with the texture of the coat and other things as well. And I looked at genes associated with high altitude adaptation in mammals, and in some cases in birds. And I looked at genes associated with the loss of spines in sticklebacks. And I started noticing there were some very obvious patterns. The genes, or sometimes they weren't actually genes that coded for proteins, sometimes they were like regulatory regions. The genes are designed so that they can change. Well, that's amazing, because you can design something that works great, but changes can destroy it. But that wasn't the case here. The genes were designed so that they could change, and the mutations seem to appear under particular conditions. Now, in the paper, my ICC paper, I explain how you can tell whether or not it's mutations or created diversity. Some of them are obviously mutations. Other times it isn't quite so clear. But these mutations seem to show up under particular conditions, whether it's domestication or when it's useful for adaptation. So years ago, I had a paper called Selecting for a Behavior and the Phenotypic Traits That Follow, and that was in the Journal of Creation. And it talked about how when you domesticate, in this case it was foxes, farm foxes, when you domesticate an animal, all of a sudden new variation shows up, which you almost never see otherwise. And also, mutations can show up in high altitude situations when they're useful for adaptation. Certain types of genes seem to be more commonly in this group that mutate. G protein coupled receptors, olfactory receptors, major histocompatibility complex genes. These are involved in helping the organism as it interacts with its environment. So the genes that are designed to be able to change are the genes where changes will help the organism as they interact with their environment. And the type of mutation varied depending on the region. So in some regions you had the VNTR, variable number tandem repeats, where you'd have a segment that was repeated a different number of times depending on the allele. In other places, it was more common to have indels or single nucleotide polymorphs, which is just a single letter change in the DNA. So there's a lot of pattern. I describe this in a lot more detail in the paper. So if you're interested, please read the paper. But based on what I saw, I had a hypothesis. Actually, it was a prediction. Many adaptive mutations are not the result of random genetic errors. Instead, much like mutations involved in antibody formation, there are enzymes and mechanisms, for example, nucleotide sequence motifs, that guide the process. Okay, I studied antibody formation when I was in vet school, and it is mutation, but it's highly directed. Now, there's random components in the program, but nevertheless, there's enzymes to initiate things and enzymes to do all sorts of things in the process. So I was saying that adaptive mutations that show up in organisms are going to be like this. I posted this on ResearchGate in our eKINDS project. And a few years later, there was confirmation of this. Monroe et al. In 2022 said, mutation bias reflects natural selection in Arabidopsis thaliana. And you can see a picture of the plant on the right. Essentially, what they found is mutations were more common where they were potentially helpful. Now, remember, they controlled for natural selection. They made it so natural selection could not be an influence there. And mutations were much less common where they can do harm. Interesting. It's almost like there's design. If it looks like natural selection, but it's not, it is evidence of God's awesome design. And that's why when people want to attribute something to natural selection, it's like, well, prove it. If you're going to claim it's natural selection, you better come up with evidence that shows that it is, and it's not something else. Well, that's great. You have a mutation and you have a new trait, and it's helpful, but only one individual has it. What's going to change the frequency of traits in a population? Well, we've been talking about genetics, but traits is a general term. Let's back up a little bit. Let's point out that physiologic and behavioral adaptation occurs too. And this is true in high altitude adaptation. It is true when it comes to blind cave fish. It's interesting because for years, evolutionists have used blind cave fish as an example of natural selection. And now we know that was wrong. And ICR has done some good work on that and published on that. And so physiologic change is something that organisms are able to do, and it's a very important part of adaptation, and it can change the traits that the organism, exhibits. The second thing is shuffling of created diversity. When I think of created diversity, I think of a work by Rob Carter. He was responding to Colin's comment that it is impossible for the human race to have come from just two people because of the diversity within the human race. And Rob Carter examined that claim and said, well, you're assuming that there was no variation in the first two humans. Every person has two copies of each chromosome. And Adam and Eve would have two. Now, we inherit one from our father and one from our mother, and they would have been created that way with just two. And the version of a gene on one chromosome can be very different from the version on the other chromosome. So you have a lot of created diversity, and you have ways to shuffle that up. And so Rob showed that, yes, you can explain much of the diversity. When you look at it on a large scale, you can explain much of the diversity by just assuming something reasonable, that human beings were created with diversity to begin with. We already talked about founder events affecting the frequency of traits in a population. But I want to finish our talk by talking about genetic mechanisms that bias what is passed on to offspring. They generally go under a broad category called meiotic drive. So my second hypothesis that I've posted on ResearchGate is mechanisms that bias allele transmission, such as biased gene conversion and other forms of meiotic drive, will eventually be shown to contribute significantly to the propagation of and fixation of adaptive alleles. I've seen design explained away a lot in my lifetime, and one way to do it is assume Mendelian inheritance is always normal and call all else intragenomic conflict or selfish, so as not to imply that there might be a purpose. And we see this all the time of talking about selfish DNA and parasitic DNA and just all this rhetoric about what's going on without looking at what's going on, because it has to be that if you have a secular worldview, because there is no God, and natural selection explains everything in adaptation, basically. Design is recognized if we can get away from that. Meiotic drive is relatively common. This is something we know. So if we look at what we know, it'll help us recognize design. The serious problems associated with it are rare, though there are examples where there are trade-offs. And I went through a number of papers where they talked about this being the Achilles heel of the genome. It was going to drive all these terrible mutations to fixation and all this. And one of the papers I discuss in my ICC paper is one that shows that, well, it looks like it more commonly drives the good alleles to fixation. So if things are as the evolutionists believe, mutations are essentially random, and the mechanism of meiotic drive is random with respect to fitness, then their prediction makes sense, but their prediction is not holding up. Okay, biased gene conversion is one example of meiotic drive. I'd like to go through it briefly. Most of you who had biology can follow this. During meiosis, which is the cell division that makes the egg and the sperm, during meiosis, there's something called homologous recombination that goes on. This is essential. This is essential for the next generation. It has to work. There's crossovers that occur, and there's gene conversions that occur. And this is a little bit overly simplistic, but it'll give you at least a basic idea. Crossovers are where two chromosomes, they're homologous chromosomes, swap segments. And that can help to scramble, shuffle things around on the DNA. And there's a double holiday junction that's characteristic of that. Gene conversions, on the other hand, are where you copy a segment from one chromosome over on to another. So you're doing a copy over rather than a swapping. Synthesis-dependent strand annealing is the model. It uses different enzymes to accomplish, but it appears to be more common. So what's going on with gene conversions? Well, you're copying one over onto the other. Now, if you copy either way equally, then you don't really change your expectations for Mendelian inheritance. Biased gene conversion is when one allele is preferentially copied over onto the other. And so, for example, if you have an organism where on one chromosome they have the capital A allele and the other chromosome they have the lower A allele or version of the gene, then when they start making gametes, if you find that it's a lot more common for one to go to the next generation than the other, one of the things that can cause that is biased gene conversion. So you see here, the big A is transmitted twice as often as the little a. So that's what biased gene conversion is. You have switched what you would expect in Mendelian inheritance, and you've biased it so that one goes to the offspring more commonly than the other. It appears to be very common. It is not associated with the harmful effects as much as predicted, not nearly as much as predicted. Creationists should be looking for purpose. This is not an accident. There's enzymes that control it. This is not an accident. On a broader scale, biased gene conversion and other forms of meiotic drive go under a broad category of transmission distortion. Basically, what's transmitted to the next generation genetically is distorted from what we would expect if Mendelian inheritance is going on. So transmission distortion is when one allele or one version of the gene in a heterozygote is preferentially transferred over the other. Centromere drive is an example. It happens in females because when a female makes an egg, you have an asymmetric meiosis. And so centromere drive affects which chromosome ends up in that egg. And I have a discussion of that in the paper. There's other systems, target killer systems or poison antidote systems can happen in males or in yeast. And I give you references if you want to follow up on that. Evolutionists paint this as an arms race. It's very interesting that you can claim there's an arms race with no intelligence involved. I find that amusing. But more than likely, just like other things in the body where there are controls that turn things on and controls that turn things off, that happens in physiology all the time, more than likely there's a purpose, and creationists should be looking for the purpose. And after looking at all that's going on, it makes me even more confident about my hypothesis or prediction that mechanisms that bias allele transmission, such as biased gene conversion and other forms of meiotic drive or transmission distortion, will eventually be shown to contribute significantly to the propagation and fixation of alleles. What does that mean? That means adaptation is not by, and diversification is not by evolutionary mechanisms. God designed mechanisms to put new variation in place, and God designed mechanisms to help them become more common when that's appropriate. So what? We have used the biblical worldview of a God who created life according to their kinds and cares for his creation, even in the midst of this fallen world. We have used that biblical worldview, and it allows for a more accurate understanding of biology, and it allows for more accurate inferences about natural history, and it highlights the manifold works of God. Psalm 104:24. O Lord, how manifold are your works. In wisdom you have made them all. The earth is full of your creatures. God is awesome.